As we've seen so far, chromosomes are pretty much the key to life. The information that's stored in a chromosome provides recipes to produce vital cellular components. The importance of this information is underscored by the fact that the cell dedicates an entire process known as mitosis to accurately separating or segregating the chromosomes into daughter cells. Now, before we learn about mitosis, let's talk some more about chromosomes. Have you been able to keep track of all this chromosome terminology we've been throwing around? Yeah, okay, me neither. So let's see if we can write everything down and sort of clarify everything in our minds before we move on. So let's see. Basically, a chromosome is a single molecule of DNA which represents a specific subset of the information in the genome. And I think we said we were going to try to remember this as each chromosome being a different kind of cookbook, right? Now, we also said that each gene is located on a specific chromosome. And we said that made sense because if we think about each gene as a recipe, each recipe is only found in a specific cookbook. Now, a diploid organism, like, say, humans, has 2 copies of each chromosome. And we call those copies homologous chromosomes or homologs. And we can think of them as kind of like backup cookbooks that might vary slightly because we have actually different editions of the same cookbook. All right, so in preparation for cell division, a cell has to make a copy of its DNA in a process known as replication. So replication yields 2 copies of every chromosome, and these are called chromatids. So it's important to note that replication doesn't produce more chromosomes. Rather, it just produces 2 copies of a given chromosome. So following replication, there are 2 chromatids associated with each chromosome. And we often refer to these chromatids as sister chromatids because they're just basically copies of the same chromosome. So really, we can just think of these sister chromatids as basically just being exact copies of the same cookbook. All right. So now that we've got all that straight, let's go ahead and introduce some new terminology. Okay, so we keep seeing this representation of a chromosome, which is all fine and well, but why are we depicting it this way? It turns out a chromosome can be divided into basically 3 different parts: the centromere, arm, and the telomere. Now, notice that the chromosome is often depicted in an X-shaped structure with a constriction in the middle. This constriction point is called the centromere. And the centromere is a physical location along the chromosome where sister chromatids are held together throughout most of cell division. And as you can see in most of the diagrams that we've used thus far, the centromere is often depicted as a circle. Also, note that the location of the centromere can vary. The centromere may be located in the middle of the chromosome, or it could be located closer to one of the ends. That makes the location of the centromere one of the traits that scientists can examine when trying to distinguish the karyotype of an organism. As we'll see later, the centromere also plays a central role in cell division. So the centromere effectively divides the linear DNA molecule into 2 parts. Now, each segment above and below the centromere is called a chromosome arm. Now, note that the length of the chromosome arm above and below the centromere varies from chromosome to chromosome. So this is also a physical characteristic which can be identified in karyotyping. The tips of the chromosomes are known as telomeres. Now, the ends of DNA are highly susceptible to degradation. And to combat this obvious problem, a special composition and arrangement of DNA at the ends of the chromosomes limits the ability of chromosomes to react with other molecules. Now, this special sequence of DNA is known as a telomere. In summary, a chromosome is a single molecule of DNA, which represents a specific subset of the information in the genome. A diploid organism has 2 copies of each chromosome, which are called homologous chromosomes or homologs. A chromatid refers to the copies of chromosomes produced during DNA replication. During mitosis, a cell could possess at most twice as many chromatids as chromosomes in its genome. A sister chromatid specifically refers to one of the 2 copies of a single chromosome. During mitosis, at any given time, there can be at most 2 sister chromatids per chromosome. The centromere is a physical location along the chromosome where sister chromatids are held together throughout most of cell division. The chromosomal arm is the portion of a chromosome found on either side of the centromere. And the telomere is the portion of the chromosome tip which protects the chromosomal DNA from degradation.